Hey, ladies and gents, you made it back for part two. If you thought metabolism and ATP was a dynamo, just wait until we talk about enzymes. All right, now enzymes. Typically, they're proteins. There are sort of a special group of molecules, ribozymes. Don't want to mention that too early, but there's stuff down the line. There are other molecules that can catalyze reactions, but typically, when we talk about catalysts and chemical reactions, they're enzymes, which are proteins. Okay. Now, what do they do? What they do is they speed up reactions without themselves being consumed. So that means enzymes sort of interact with other molecules reversibly. The enzyme itself can catalyze reactions over and over and over again. Under optimal conditions, one enzyme can catalyze 400 to 500,000 reactions per second. So enzymes are incredibly effective at speeding up rates of chemical reactions. Now, how do they do that? They do that by lowering activation energy. It's the energy needed for a chemical reaction to occur. Spontaneous reactions can occur. Bonds can break, energy can be released, but typically it may take a long time for that to happen. So activation energy is the energy you need to put into the system in order to get the reaction to occur, in order to get those bonds to start breaking. Think of we've talked about firewood, right? There's a lot of potential energy in firewood. Well, firewood will burn and release a lot of energy. It's an exergonic reaction, but you have to put some heat into it, right? So activation energy is the energy you have to put into the system in order to get those bonds to start breaking and energy be released, okay? So if you look at the graph that you see here, you see the energy in the reactants. That's molecules A and B. And then you see them joined together as the products. Well, activation energy is what you have to put in to the system before the reaction occurs. And you can see that with an enzyme, the activation energy, the energy you have to put into the system is much, much lower. I guess if you really like frogs, you can look at that as the energy needed to get those frogs to have a big frog party there. Party in the pond. Okay, now activation energy, again, it's the energy needed to get the chemical reaction started. So you can think of like gasoline. You can release a lot of energy in gasoline, right? But you'll have to apply a spark. You have to apply some energy to get those bonds breaking. Okay. Now, enzymes can lower activation energy in some interesting ways. Imagine I had like a meter stick and I asked the person to like karate chop that and snap it in half. Okay, they could do that, right? They could take, imagine my hands are molecules and the meter stick is the bond between them. Let's think of it that way. If my hands are individual molecules in like a polypeptide, and the ruler is the bond between them, if you go and try to snap that ruler, you could probably do it, but it's going to take some energy. But what if I bent the ruler and put a lot of stress on that ruler? If I put a lot of stress on the bond, is it going to take as much energy to break the bond? Well, no. So that's one thing that enzymes can do, is they can actually take a molecule and twist the molecule so that it stresses the bonds of the substrate. And if you stress the bonds of the substrate, it's going to take less energy to break the bond, and that's going to make the reaction happen faster. So that's what they indicate by this transition state here that you see between these molecules. You can take an original molecule, stress the bonds between the atoms or between the molecular subunits, and then it takes less energy to break the bonds, so the reaction happens faster. So that's one cool thing that enzymes can do. Okay, now why are enzymes so critical to life? Again, one enzyme can catalyze hundreds of thousands of reactions a second, so it really speeds up the rate of reactions. Additionally, it does that without the addition of heat, right? So you can imagine that heat breaks bonds. So you could break bonds pretty readily with heat, but the problem is if you heat up the body too much, well, then you're going to break the bonds of your own molecules. So running at really high temperatures isn't really compatible with life. So what enzymes do is allow chemical reactions to work quickly enough at lower temperatures that are more supportive of life. Okay. Another cool feature of enzymes is that they have a high degree of specificity, right? The active site of enzymes has a really particular shape. We've talked about the lock and key and induced fit notions or models. So if you have certain enzymes present in certain organelles or in certain cells, they will catalyze different reactions. So now think about, a eukaryotic cell, if you have all these different organelles with different enzymes in them, you could have certain chemical reactions concentrated, and then you can become much more efficient at creating new molecules. So by having different combinations of enzymes, different organelles, or different cells, you can do different reactions, and you can start to control the rearrangement of matter in life or in cells. Okay. Now, one thing that's really important to note, and I've seen questions related to this on the AP exam before, an enzyme speeds up a reaction, right? It makes it happen faster, but it doesn't change the overall progress of the reaction. When you think about the delta G, the conversion of reactants to products, it's going to either be a positive or a negative delta G. It doesn't change the overall amount of energy. It just lowers the activation energy. So if we look at this graph, here in red, you have the enzyme catalyzed reaction with the lower activation energy. Here is the uncatalyzed reaction in blue, has a higher activation energy. But look at the overall change in delta G. The reactants had more energy than products, right? But would the delta G have changed at all between reactants and products? No. It's still releasing the same amount of energy. It just took the input of less energy to get that going. Now, with all these graphs, I do have a little bit of a issue with how they draw things out because they show the change in activation energy, but at lower activation energy, this should also be happening sooner, right? So the progress of the reaction should ideally be occurring sooner. Okay, let's get or discuss the parts of an enzyme. Again, the active site's important because that's where the enzyme interacts with the substrate. So that's where the change occurs. So active sites have really precise shapes because there are certain amino acids in those active sites with different chemical properties. So each active site in enzymes is going to have its own sort of unique three-dimensional shape. Now, again, we've talked about the induced fit model. The idea is that when reactants interact with the substrate, it's sort of like a handshake. The substrate will change its shape, or the reactant will, when the reactants or the substrates interact with the active site of the enzyme's active site may actually change its shape a little bit to more closely interact with the substrates. So if you look at this model, the induced fit model here, you'll notice that here are the substrates with very precise shapes, and the enzyme's active site isn't an exact match. But when they interact with one another, the enzyme changes the shape of its active site slightly to more closely interact with the substrate. Okay. And of course, these are reversible reactions. So the products could interact with the active site of the enzyme and then get converted back into reactants. So that's why you have whatever product you have in a chemical reaction, you better do something with it. You use it in some other metabolic reaction or get it out of the body. Okay, so how do enzymes actually speed up reactions? They do that in different ways. One is they can act sort of like a template. So you can organize the substrates. So you can imagine if you have all these different molecules moving around in the cell, for one molecule to bump up with another molecule in just the right orientation, that probably doesn't happen very often randomly. But what an enzyme can do is grab one molecule and hold it, and then it takes the other molecule and brings it up in the appropriate conformation or orientation so that they can interact with one another appropriately and cause a chemical reaction. Okay, so this idea of organizing substrates, it's basically like a template where you fit one piece of the puzzle in, and then you fit the other piece of the puzzle in, and then they can interact with one another and form chemical bonds. Okay, enzymes can create these little microenvironments. So again, there's like a three-dimensional shape to the active site, and what that can do is maybe have some R groups of the amino acids be acidic or basic. So you can actually have an active site that has a little microenvironment that has a slightly different pH than the surrounding cytoplasm in the cell. And so that can allow chemical reactions to occur. Again, enzymes can stress the bonds of substrates. Again, if you take substrates and bend them or twist them into that transition state, it takes a lot less energy to break the bond, so it happens faster. Okay. And again, you can sometimes have the R groups of the enzyme interact directly with the substrate, break the bonds of the substrate, and then the molecules in the substrate can interact with other molecules in different ways. Okay, now this is, again, examples of stuff they like to get into on the AP exam. What can influence the rate of enzymatic reactions? All enzymes have an optimal pH and an optimal temperature. Okay. So our body temperature is 37 degrees Celsius. Okay. So we run at like 37 degrees Celsius. The enzymes in our body have evolved to operate best at around 37 degrees Celsius. If those enzymes deviate from that temperature too much, what do you think is going to happen to reaction rates? Here goes down. Enzymes also work at optimal pH. Okay. Enzymes that are found in the stomach will work best at a pH 2. Enzymes that are in other body fluids or inside of the cytoplasm of cells, they operate at a different pH. There's a cool example from the previous test, right? The enzymes and lysosomes work best at a pH 5, and then the cytoplasm is pretty close to pH 7. So for the hydrolytic enzymes for a lysosome to work, you have to pump protons into the lysosome to lower the pH so the enzymes become active and can digest, old cell parts or pathogens or whatever. Another molecule, or not even molecule, another substance that can change the rate of chemical reaction are coenzymes, or sometimes called cofactors. Coenzymes or cofactors speed up reactions by sort of helping enzymes along with their job. This is what vitamins can do as well. So you think about how vitamins are useful to us. So if we look at this protein, the blue protein here, and you look at the substrates, L1 and L2, and they're probably not going to fit on this protein particularly well. But the cofactor or coenzyme, I think of it as being like a saddle on a horse. The cofactor or coenzyme can fit into that active site, and then the enzyme becomes active and you can catalyze the reaction. So vitamins are cofactors, coenzymes. These are molecules that interact with enzymes, and when they combine, you notice how they change their shape, and they can speed up chemical reactions. What else can affect enzymes? Again, these are really common concepts to look at on the AP exam. Competitive inhibition. Okay. When we think about competitive inhibition, here's the substrate, there's the active site. Notice that they're fairly complementary in their shapes. Well, what if you had some other molecule, like a red molecule, that is similarly shaped to this blue substrate? If they're similarly shaped, could the red molecule sort of occupy part of that space in the active site and block the blue molecule? Well, sure. So a competitive inhibitor will actually compete for access to the active site with the natural substrate. Well, you can imagine that if you add a competitive inhibitor, reaction rates are going to go down because the active site of the enzyme is occupied more frequently by this competitive inhibitor. Now, the application questions they could ask on the AP exam, well, how do you know if it's a competitive inhibitor? Well, let's imagine a ratio. If it's a 1-to-1 ratio of like blue substrate to red substrate, which is the competitive inhibitor, what happens if you add more of the blue substrate? And now it's 5-to-1 blue to red. If it's 5-to-1 blue to red, then you would expect that reaction rates would go up, right? So let's sort of walk that back. Let's say you only have the blue substrate. And if you add the blue substrate, the enzyme catalyzes the reaction and you have some known reaction rate. If you add the red substrate as a competitive inhibitor, the reaction rate should drop. But then if you change the ratio and add more of that blue substrate, then the reaction rate should go back up. Because again, it's just greater odds that the blue substrate will interact with the active site than the red substrate. Now, a non-competitive inhibitor, it's a different situation. A competitive inhibitor competes for access to the active site, but a non-competitive inhibitor, looky here, it docks somewhere other than the active site. But look at what it does to the shape of the active site. So you see here, this sort of juts out. Oh, nope, shuts it down. Okay. So with a non-competitive inhibitor, it doesn't compete for access to the active site. It docks somewhere else and changes the shape of the active site. So now, what if we change the ratios again? If you add a lot more of the blue substrate, if you're using a non-competitive inhibitor, is the reaction rate going to go up? Well, no, because the non-competitive inhibitor is going to literally change the shape of the active site. So it doesn't matter how much of this blue substrate you have, you're not going to be able to bring the reaction rate back up. So that's how they tend to approach things on the AP exam. Like if they give you some scenario and they want you to figure out if some molecule is a competitive inhibitor or a non-competitive inhibitor. If it's a competitive inhibitor, changing the ratios can change the reaction rate just because of the odds of the particular substance docking on the active site. But with non-competitive inhibitors, it doesn't matter what the ratio is or how much more substrate you add, you've changed the shape of the active site, so you're not going to increase the rate of the reaction. Okay. Let's talk briefly about one interesting example of a competitive inhibitor. It's myostatin. Now, the prefix myo means muscle, and stasis means standing or staying the same. So myostatin is actually a molecule that docks on receptors on our skeletal muscles, and it tells them, stop adding protein, stop growing those muscles. So we actually have a molecule that inhibits our muscle development, our muscle growth. Now, why would the body do that? Well, it's because muscles are real energy hogs. They require a lot of ATP, and they require a lot of protein. So should your body be investing in proteins and ATP if it doesn't need to? Well, no. So what myostatin does is it sort of puts the brakes on our muscle development so we don't overinvest in muscles. Okay. Now, myostatin is a competitive inhibitor. What if you blocked myostatin? If you block myostatin, then would the body get the signal to stop dividing? No. So here you can see myostatin, and then there are myostatin inhibitors. And when that happens, then muscles can continue to grow and divide. And this is seen in Belgian blue bulls. Belgian blue bulls, they don't use myostatin blockers, but what happens is they don't produce myostatin. And because they don't produce myostatin, they don't have those brakes on muscle development. So let's look at what one of these looks like. So that is what these animals resemble. So with myostatin, if you don't block or if you don't produce myostatin, you don't have that inhibition of muscle growth. And so you get these animals with these massive skeletal muscles. Now, one thing that's really interesting with this is there were some mice that just came back from space not too long ago, and the mice were up on the space station with an experiment. And what they were doing were using myostatin blockers on the mice. And what they found is that mice on the space station who were given myostatin blockers without even exercising developed larger muscles. The idea is if you're going to have people spend extended amounts of time in space under low gravity or zero gravity, could having something like this myostatin blocking drug help with maintaining muscle mass? Because, if you don't, I think astronauts have to spend like 2 hours a day exercising. They have they're strapped to treadmills and they're strapped to exercise bikes, and they have to exercise a couple hours a day to try and slow muscle mass and bone loss as much as they can. But it's still inevitable in space under zero gravity. So they're hoping that with myostatin drugs or myostatin blocking drugs, you can produce more muscle, for people spending extended amounts of time in space. Or the idea is for people who are aging or people who have muscle atrophy diseases, they may be able to get treatment for that. All right. Let's see. Now, when you have these non-competitive inhibitors here, docking somewhere other than the active site, this is called allosteric regulation. The prefix allo. Oops. Sorry, my wife and daughter just got home, so the dogs are excited about that. And my son is too. So allosteric regulation. Allo means other. So with allosteric regulation, that means a molecule docks somewhere other than the active site, changes the shape of the active site, and then switches off the enzyme. So here in part B, inhibition, there's the substrate with sort of the brownish green color looking thing there. And then here is the non-competitive inhibitor. again, it docks at this allosteric site, and then look at what happens to the shape of the active site. It changes, right? So you shut off the enzyme. And now this is significant because this is how a lot of your negative feedback works. So you have chemical reactions, right? You have enzymes that convert molecule A to B, and then another enzyme converts B to C, and then a third enzyme converts C to D. Well, what happens is molecule D can actually loop back and inhibit that first enzyme in the pathway or an earlier enzyme in the pathway. So if some molecule at the end of a metabolic pathway will inhibit an enzyme earlier in the pathway, that shuts down its own production, right? That way you don't overproduce a molecule. So allosteric regulation is really important because it's our primary means of negative feedback. Molecules we produce will inhibit enzymes earlier in the pathway so you don't overproduce those molecules. Boom. So there we have it. Enzymes. If you have any questions, let me know. Otherwise, what I ask that you do, take a minute, summarize these big ideas from the lecture. Okay? So summarize the big ideas. That'll be the first thing you review when you take your notes. And I applaud you for making it through this. Thanks so much.